First, I will give a quick introduction about Tania Patiño. She is a PhD candidate who works in the Institute for Bioengineering of Catalonia, whose professor is Sanchez, and her topic will be swarming behavior and in vivo monitoring of enzymatic nanomotors within the bladder. So let's begin.
Okay, so now I can share the screen, I guess?
Yes, you can share.
Okay, let me check.
Excuse me, I need to move to the co-host.
I don't have the permission, it tells me.
Okay, I think now you can share your screen.
Okay, yeah. Okay, can you see?
Yes, yes, I can see you.
Okay, now. Okay, so yeah, I will start talking a bit about our motivation to do this work. So during the past years, we have seen that in nanomedicine, there has been a lot of works trying to investigate new types of nanocarrier to bring drugs wherever we need them to be in the body. However, there is a lack of efficiency in these systems, and a study in 2016 revealed that only 0.7% of these newly developed carriers are able to reach their targets in vivo. This means that we need to improve these systems, and mainly the challenges that they find is that in our body we have many biological barriers that we need to overcome before we can bring these nanocarriers to our target location. So in our group, during the last years, we have been thinking about changing these systems and providing them with self-propulsion. So maybe with if the particle is self-propelled, it will be more efficient in crossing these biological barriers, and it would be more efficient in reaching their targets. So what we have been doing so far is to build enzyme-powered nanomachines. So these are micro nanomotors that are able to move using the chemical energy by enzyme catalysis. So we have been focusing mainly on urea, but we have also explored different types of enzymes, studying what are the properties of enzymes that make a nanosystem efficient to move. We have studied also how these particles move. Here, I will show you an example of in this video, you can see the control particle, which is moving, which is displaying mainly Brownian motion. And in this case, in the right, you can see a particle which is consuming urea thanks to urease catalysis, and it's moving in a very nice and directional ballistic motion. We have also done some studies on how these enzymes are located in the particle, because we need some sort of asymmetry in our system to make the particle move. And only by putting the enzymes in contact with functionalizing the particle with enzymes, we have seen that there is an intrinsic asymmetry that allows them to move. We have been also investigated how we can use these self-propelled systems to deliver drugs in 2D cell cultures, also in 3D bladder cancer spheroids, and we have seen that if we have a particle that is active, it will be more efficient in delivering a drug and also on targeting 3D spheroids. And now we are moving towards in vivo applications, and this is the work that I will be focusing on today. So the main question that we want to answer in this work is, how can we see these catalytic micro nano robots once they are inside the body, and whether these will be able to move inside the body, because of course they will find very complex environments, and this is something that needs to be studied so we can see whether this has potential for biomedical applications or not. So in the literature, there have been different examples of using medical imaging techniques to follow the micro nanorobots ex vivo and in vivo. So here I put you some examples. The first one, they were using fluorescence to monitor these micro nanorobots in vivo and also ex vivo, also SPECT and using different types of imaging also in vitro, OCT, so different examples you can see. In our case, what we wanted to do is to use PET-CT. This is a very common medical imaging technique mainly for diagnostics in cancer and many other diseases. We did this work in collaboration with Dr. Jordi Llop at TIC Biomagune, also located in Spain. And this technique, the nice thing that we wanted to exploit is that it is composed of two different imaging techniques. So first, we have the computed tomography, and this technique is based on using X-rays to obtain morphological information of the body, of the tissues, organs, and you can have a very nice 3D reconstruction of the whole body. But this doesn't give us functional information. We don't know whether these organs are active or not, and whether they have some metabolic change. Instead, we can use positron emission tomography. This is based on the use of radio-labeled molecules that will accumulate in certain parts of the body or organs, and it will give us information of how active is the organ. And this is very useful in biomedicine because we can have both types. With combining both imaging techniques, we can have a lot of information of what's going on in the body. So we were interested in using this to visualize our micro nanorobots. Another interesting thing is that the techniques I mentioned that have been used before, they have a limitation on the penetration depth, so we cannot image parts that are very deep in the body. But with this technique, we don't have this limitation because we are using radio X-rays and also radioactive material. So we eliminate this limitation. So as I said, this technique requires of using radioactive molecules. So the first thing we needed to do was to synthesize nanobots that we can radio label. For this, we used two different strategies. On the first one, we used a radioisotope bound directly on the enzyme. And on another strategy, we used gold nanoparticles that we could also radio label. We did some characterization of the resulting particles. Here you have a TEM imaging where you can see the silica, mesoporous silica nanoparticle with the gold nanoparticles on the surface that will be used later for the radiolabeling. Here is a characterization of the radiolabeling efficiency using both strategies. Then the first thing we also wanted to explore is normally we are used to track single particles and study the movement of those particles. But if we think of putting this in the body and using them as efficient delivery carriers, we will need millions of them. So we also need to understand how these millions of particles can move. So first of all, we put them in vitro under an optical microscope in the absence and the presence of their fuel, and this is what we could observe. So when there is no fuel, we observe that the drug is very homogeneous and rapidly sediments on the glass slides. Instead, when we have their fuel, these particles start to move in the form of swarms. And as you can see, they have a very different distribution from the control. In the end, they will, some of them with sediment, but initially the movement was very different. We characterize this type of swarming behavior using different imaging post-processing techniques, and which help us to visualize how these are moving in the form of swarms. Then once we observed this type of motion behavior, we moved to using PET-CT also in vitro. So for this, we designed different type of phantoms with different geometrical shapes, and we image them using PET. Using PET. So here you can see a video. On the left side, we have the nanobots without the fuel. On the right side, we have the nanobots in the presence of fuel, so they are active. As you can see, in all the cases, regardless of the geometrical shape, they were able to reach the end of the phantom in the presence of the fuel. Here you can see this is the most clear example when we have a straight line. So the nanorobots are able to move in the form of swarm and reach more efficiently at the end of the phantom when they are active compared to the control ones. Here is another example of using some more complex shapes in case that these nanobots need to travel in confined environments. So you can see here that they are also able to reach the target location much more efficiently when they are active. We can do some quantification of this of the radioactivity. So here you can, in this figure, what we did was to divide the phantom in different regions. So for each color representing one region, we can have a very nice characterization of the radioactivity. So this helps us to quantify the movement of these robots along the track, which is very useful. The next step was to inject the nanorobots inside a mice and see if we were able to monitor them. In this case, we started by doing an intravenous injection because we wanted to observe what is the biodistribution of the nanorobots once they enter to the bloodstream. And as you can see here in these images and also in this video, the nanorobots accumulated very fast, mostly in the liver. So we did a study characterizing the radioactivity in different organs, and it was very much accumulated in the liver. So this means that if we want these nanorobots to go somewhere else, we need to change the strategy, because otherwise they will accumulate in this organ very rapidly. And also this study helped us to identify that using fluorine was the best radiolabeling strategy because it was more stable. And in the other case, in the case of iodine, we can see that it accumulates in the thyroid. So we selected the fluorine from now on for the following experiments. As I said, if we inject the nanorobots, it might go to places where we don't want them to go. In our group, we have been working mainly on urease nanorobots, and the main application that we are exploring is to use them for bladder therapy. Why? Because obviously, where the part of the body where we have more urea is in the bladder, and then we thought that maybe this could be a very good application for our system. And what we want to do here is to replicate the protocol that is done currently in the clinics. So in the clinics, if they want the chemotherapy to go to the bladder, they will not inject into the bloodstream, but directly inject through the urethra. So we can have a direct injection in the body. So it's a very localized administration. We did this with our mice. So what we intravesically injected the nanorobots, and here is what we found. So here you can see an image of the bladder of living mice obtained with PET-CT. And once we administered the nanomotors in the presence of urea, initially they start forming different phases, but at the end there is a very homogeneous distribution. You can see it better here. In the case of control, what we observe is that there are two phases formed. I will show you better in the quantification. So here you have all the images of this of the bladder of the mice with the different conditions. So we tested in the presence of urea and also in the absence of the control. And we tested nanomotors and also VSA-coated nanoparticles as a control. So these particles don't have urease, they will not catalyze urea, so they are not active. So in all cases, what we observed was that in the end, so after 45 minutes, there are different phases formed in the bladder, but in the case of active nanomotors, it seems that their distribution is more homogeneous. So we quantified by this effect by monitoring the radioactivity percentage in different regions of the bladder. So here you can see that initially when we have nanomotors in the presence of fuel, initially they will form these two different phases, but in the end, these phases are homogenized, as you can see here. In the case of the control, instead what we see is that once we inject the nanorobots, but without urea, they will start forming two different phases. So it means that the particles, what we hypothesize is that the particles are not moving. So upon the entry of new urine in the bladder of the mice, these are displaced. In the other case, we have the particles that are active, moving, enhancing the fluid mixing, and that's why in the end we have more homogeneous distribution. Here, we also characterize the same effect using the control particles, so without urease and BSA, and we saw the same effect as for the passive nanomotors, meaning that only when they are active, they will homogenize the fluid and distribute more homogeneous within the whole bladder. So, yeah, as a conclusion, we observed that if we put many urease nanomotors together, they will form this sort of swarming behavior that we can monitor using simply optical imaging techniques. Also, we have demonstrated that using PET-CT, which is a very common and useful technique in medical diagnostics, is suitable for tracking the motion of these nanorobots, and that once we inject active nanoparticles inside the organism, in this case inside the bladder, they enhance might enhance the fluid mixing, and they result in a more homogeneous distribution, which could be very helpful to deliver drugs to the whole bladder, and also to reach more efficiently the target location, compared when we have passive particles where they will accumulate in one part and maybe not be efficient in reaching where they need to be. So this is all from my side. I would like to thank the group from Professor Samuel Sanchez, our collaborators that did all help with all the imaging and mice experiments, and my current group, and of course all of you for your attention.
Okay, Tania, thank you for your wonderful talk. Now let's move on to the Q&A session. If anyone has questions, just unmute yourself and speak out to your questions, or you can text in the chat box, and Tania will answer the question for us.
Hola, Tania. Can you hear me?
Hmm?
Hola, ¿cómo estás?
Hola.
Thanks for your nice talk. This is Lei. Actually, very nice talk and very nice work. I have one question, please. The first one actually is, why does this nanomotor move in a small style?
Why they move in a form of swarm is your question?
Yes, because comparing your slides, especially the last one showing the zero concentration of the urea and 300 micromolar, right? Micromolar of the urea. There is a swarm formed when they move. So that's why.
So your question is why they move?
Yeah, you can see the last one, the 20s, right? The first image on the left side. Yes, this one.
This one?
No. Next one, please. Next slide. Next one. Yes, this one, exactly. The swarming behavior.
Yeah, so the thing is that in this case, the swarming behavior we observe when we have a drop and we are observing it with our microscope, we observe this type of course, this is a simpler media. In this case, it's more complex, it's inside the body, so we don't have the resolution to observe the small nanoparticles. So here we observe this cloud of radioactivity, so we can observe the same swarms that we were observing with the optical microscope. In this case, what we hypothesize that is happening is that because the particles are active, maybe they are doing this swarming that we cannot observe with this technique, but maybe it's also happening. And in the end, the whole radioactivity is more spread and it's more homogeneous. And in this case, it doesn't mean that it is a swarm, but it's that the particles are accumulated in one side of the bladder, because the mice, when it's alive, so it will keep injecting liquid in the bladder, because it's the urine coming from the kidneys, and it's entering. So we hypothesize that this liquid is displacing the particles. So not that they are swarming there. I don't know if this answers your question.
Yes, yes. I still cannot understand why when there is no urea, it is distributed very normally or evenly. But when we add some of the like 300 millimolar of the urea, there are some kind of swarm or some special pattern existing there. So why it does just the more frequently or more uniformly distributed? That's my question. Right?
Yeah, to us it was also very surprising in the beginning because when we put the drops, as you can see, so in the optical, let me show you. Here. So here, when we put at this small time, we put the drop, right? Are you seeing the video? So these ones are like actively moving and forming these swarms. The effect that we are seeing with the PET-CT imaging is different because there the imaging is the whole radioactivity. So we are not seeing the particles, we are seeing the whole cloud. So we think that in the beginning there is this sort of two-phase formation, but in the end is more homogenized because the particles are moving everywhere. And in the end is everything spread and the fluid is mixing and everything is mixed together. In the case of the passive particles, they are accumulating because the liquid is entering and displacing them, but not because they are actively swarming. You know what I mean? In the case of
very nice. Yes. Yes. Okay. Okay. One more question, please. Actually, I was also thinking when the nanomotors were injected into the bladder, do they also follow this kind of moving rule, or do we have any proof on their motion behavior? Because we cannot see them clearly anymore, right? Within the bladder is too hard.
Exactly. So what we can see is what is this type of images is the best we can see. So it's like the whole, but we can quantify the radioactivity in different parts of the bladder. So we can know if they are accumulating in one part or if they are spread and distributed. This is something we can do with this technique. Of course, we cannot see very small particles as in the case of the optical microscope because we don't have this resolution yet. But we can have these measures by measuring the radioactivity at different points of this bladder or this cloud that is formed. So I think that it's a bit different, but it's useful for that.
Yes. Okay. Muchas gracias.
You're welcome.
Tania, I saw that there's a question in the chat box. Can you read it, or I can read it for you?
I can read it, yes.
Okay.
So why do you need to add urea to the bladder? Because it's already there. So the question, the thing is that normally the procedure is that the bladder needs to be emptied, cleaned, and this is what they do in the clinics, and then they put the treatment. We wanted to do the same as in the clinics. And another reason is because in the bladder there is urea, but we don't know the concentration because it might vary from animal to animal, person to person. It's very heterogeneous, different moments of the time. We wanted to have a systematic study and know exactly which is the concentration we are using. So this is the procedure. We empty it and then we inject liquid, but fully controlled. And then of course the animal will be putting more urine with urea, of course. And this is why we see this effect that I was talking about.
Okay, thank you for your answer. Any questions from the audience? Let's wait for another 20 seconds and then we will.
Tania,
Okay.
this is Wei from Shenzhen. Hi, can you hear me, Tania?
Yeah, very low, but yeah.
Okay, all right, sorry. Let me try to speak up. I guess I wonder if anyone from your lab or outside of your lab is following up on the swarm dynamics or some mechanism behind the swarming? I know Lei was asking the same question, but are you guys pushing forward on this front?
Yeah, I think that in the group, Samuel is very interested in this behavior because it was for us very nice, but also surprising. And we would like to understand what are the interactions between different particles that are making them move in the form of swarm, because then we can better understand what's happening and better control our systems. This is something that, yeah, we are interested in looking at what type of interactions are happening. And yeah, I guess it will be a follow-up.
The observation reminds me of the recent Nature Nanotechnology paper from Jinyao Tang from Hong Kong University. I don't know if you read it, but they also reported some sort of swarming behavior with, I remember, zinc oxide nanoparticles. I know it's a very different system, probably different reactions are happening, but maybe, but I remember in that paper, there's very nice simulation that helps with understanding. Maybe you guys can do the same thing. Yeah, definitely. We need to think about this. Of course, it's a different system because we are producing different products, electrochemical and hydrodynamic interactions might be different, but yeah, it's totally something that we want to take a look at. And yeah, maybe this paper is an inspiration. I look forward to it.
Hello, can I ask a question? Great, really nice talk. I think it's wonderful imaging, and it's nice to be able to see these kind of things used in vivo environments, which has been a very hard task for most nanomotors. So congratulations, this is a nice result. The question I have is the one motivation that you started with, it matches really well with the kind of things that you're starting to show, how you can image things inside and how you can try to see where they're going. Is there a way, do you think, that you could start targeting really, like what you were saying, that very few drugs actually are going towards the target? That's one of the great advantages of a nanomotor that actually is moving. So is there some way that you can couple these to a targeting sequence so that you actually will be able to get from the random movement to where you actually want to go?
Yeah, so far we have tried with coupling antibodies on the surface of the motors and seeing whether having this recognition element can improve the targeting. We have seen that if we have both, if we have the targeting antibody and also motion, the targeting is much more efficient. But we have tried this in 3D bladder cancer spheroids in vitro. So the next step would be to check this, if this is also happening in vivo.
I see. And the kind of targeting, I mean, is the things that is in spheroids something that you've published already? Yes.
So it's a ICS Nano paper publication already. Yeah, we were exploring this. So bladder cancer cells express some proteins on their surface, and we were targeting these proteins using specific antibodies. And this can be tuned on demand. We can use any antibody, and we could eventually try to target different parts of the body with this and help with the motion.
I see. So in theory, I guess you don't see any reason why the same type of targeting wouldn't work in the bladder kind of case.
Well, in vivo, it has to be studied because when we go in vivo, there are many things that will influence this targeting. So maybe there can be some proteins that can attach to the surface and modify. So in principle, could work, but of course it's something that we need to study in depth because moving in vivo, it's adding much more complexity to the environment, and we need to start learning how to control these parameters and how to overcome these barriers that I was talking in the beginning.
Right. That is great. Yeah. if I could just follow up, may I ask how expensive are these kind of enzymes to make right now? If you're trying to make something that is of a decent kind of amount for a type of treatment.
To make the enzymes, so right now we have been using commercial enzymes which are not too expensive. So this urea is also the particles are not expensive at all to make, and we think that this could be scaled up. To make them, I am not sure how much will it cost, but I guess it could be done. Not too much, I guess. I cannot give you a number. Sorry.
Thank you very much.
Thank you.
Any other questions from the audience? Let's wait for another 10 seconds. Okay,